people. So, yeah. Hi, all. Good evening. Good evening, all. I was new for this section. Okay. So, yeah, it's fine. You can find all the three TA for this course. Three TAs are there, and everyone recorded video you can find in your NPTEL module in the left side. So, all the links are there. Today I'll speak about the biomolecules. So today I'll speak about carbohydrate, your lipid and fatty acid and nucleic acid. Next class we'll speak about or we will discuss about the proteins. So, okay. I hope I'm audible and I'm visible as well. Okay. So what I'll do, I'll share my screen and then we'll start. Okay. So can anyone just quickly confirm if the PPT is visible? I think it is, yeah, it is visible, right? I'll go to the chat box. Okay. So yeah, just anyone can quickly confirm if the PPT is visible to you people also. Yeah, okay, great. Um, we'll start today's session then. I think most of the people are here then. Okay. Okay, great. So, good evening, all. Welcome to today's week 5 problem solving session. I'm Rajesh. I'm a PMRF research scholar at the Department of Biotechnology in IIT Madras. And as I said, today I'll speak about glycolysis. Discuss about the biomolecules. And these biomolecule concepts, this is what you have already studied in most of these things you have already studied in your chemistry classes. If you remember last time, I gave you some brief about electron transport chain, and I said we will discuss more about electron transport chain in the later class. So today you'll also look at the electron transport chain or ETS, electron transport system as well, because glycolysis is followed by electron transport system to get the full output or get the full output of energy. So the first thing we'll be discussing is glycolysis from which is kind of a continuation from the last week. So let's start with that concept of glycolysis. So glycolysis is a Greek word. So which means glycos stands for sugar and lysis means splitting. So here sugar is getting split. And the scheme of the glycolysis was given by three scientists, Gustav Emden, Otto Meyerhof, and J. Parnas. And that is the reason why this is always uh, as well referred to as EMP pathway for Emden, Meyerhof, and Parnas pathway, EMP pathway. So, yeah, like a glycolysis and EMP pathway, both are same. Don't get confused in this. The main thing to remember that glycolysis occurs in the cytoplasm of the cell. In every cell, glycolysis occurs in the cytoplasm, not anywhere else. So the glucose is split in the cytoplasm and is present in all living organism because all living organism needs energy, and if they need energy they have to produce energy, and glycolysis is the energy producing mechanism. So in this process, glucose undergoes partial oxidation to form two molecules of pyruvic acid. I think you know about this glycolysis step and the enzyme involved. And in the end, the six carbon glucose. So you know, glucose is a six carbon molecule. Also, we'll have a look at the in the upcoming slide. But the six carbon glucose is split into three carbon pyruvic acid, two three carbon pyruvic acid. So on the left, I have put that glycolysis pathway and the enzyme involved in each step. So there is total 10 step in glycolysis, and there is total 10 enzymes involved in the each step. So starts with the phosphorylation, like glucose 6-phosphate, fructose 6-phosphate. And what I would like you to do is you note down in which step ATP is produced and in which step ATP is consumed. In the first step, ATP is consumed. See, ATP here is consumed because this ATP, adenosine triphosphate, is donating one phosphate to become adenosine diphosphate, so that the glucose can become glucose 6-phosphate. So there is here one loss of ATPs here. Same thing for here also, we are having a loss of ATP. But in this step, further towards the fructose 1,6-phosphate, we get the dihydroxyacetone phosphate or DHAP and glyceraldehyde 3-phosphate or G3EP. Here, simultaneously, like this can kind of interchange between each of this. Okay, so that is the reason why from this here onwards everything is twice. So uh, you can think of this as a, like six carbon is split into three carbon and three carbon here. So everything becomes twice. Note here we have two ATP gain here. Two ATPs produced in this step of 1,3-bisphosphoglycerate to 3-phosphoglycerate. And the step where phosphoenol pyruvate goes to pyruvate, we have a net gain of ATP, 2 ATP. So 2 to 4, total gain of 4 ATP, out of which we lost 2 ATP. So net become 2 ATP. Along with this ATP, so the main objective of glycolysis or your electron transport chain is the production of ATP, because ATP is the currency of energy in the cell. So note here, along with ATP, you are also getting NADH2, NADH2, and of course, the final product is pyruvate. So if someone asks, the question is there, what is the end product or the net product of glycolysis? That is two molecules of ATP, two molecules of NADH, and two molecules of pyruvate. We will see how this NAD, like ATP is understood, it is the currency of your energy. We will see how NADH is going to produce more ATP, and we will see what is the role of pyruvate exactly. So for aerobic respiration, so we are talking about, uh, I'm 10th class student and this is all present in my textbook. Yeah, uh, this course is actually a undergraduate first year level course. So most of the things you have already studied in your 10th, 11th, and 12th. These are not very difficult things or not very advanced thing. This is a UG level, undergraduate level course. Okay. So I'm currently talking about aerobic respiration because aerobic respiration produces much more energy or ATP or energy as compared to the anaerobic respiration, that also we will compare. So aerobic respiration, we are currently talking about glycolysis, take place within the mitochondria. So yeah, last time I told you about the mitochondria. I told you there is two membrane present in the mitochondria, and I showed the how hydrogen or H plus ions are like concentrated in the mitochondrial membrane, in the intermitochondrial membrane, and this H plus ion cross the F0, F1 particle or the ATP synthase, and during that process, ATP is produced. So that is the final step of your aerobic respiration, which occurs in the mitochondria. So, if the final step of respiration, aerobic respiration, occurs in the mitochondria, this pyruvate, whatever is produced in the cytoplasm, has to go to the mitochondria. So, that is the step written here, that pyruvate is transported from the cytoplasm into the mitochondria. So remember these two crucial event in the aerobic respiration. The first is the complete oxidation. The pyruvate will undergo oxidation by stepwise removal of all hydrogen atoms, leaving molecules of carbon dioxide. How many carbon dioxide will you get? You will get three molecules of carbon dioxide. That also we will see. And this particular event occurs in the matrix of the mitochondria. So, if you remember, if this is mitochondria, and this is like the double layer, and this is the matrix of the mitochondria, and this is the inner membrane, this is the outer membrane, this is the outer membrane, and this is the inner membrane. Okay. So please have a look. If you don't remember the structure of mitochondria, just have a look. So the first happens at the matrix of the mitochondria. Then what will happen? The passing of the electron removed as a part of the hydrogen atoms to the molecular oxygen with simultaneously synthesis of ATP. And this is the location is inner membrane of the mitochondria. Okay, I'll discuss with this later. I'll share everything. I'll tell you in details after the class ends. Okay, you just focus on this for now. So, uh, so these are the two crucial events: what complete oxidation of the pyruvate and the passing of the electron removed as a part of hydrogen atom to the molecular oxygen simultaneous synthesis of ATP. So this is the electron transport chain we are talking about, and the final acceptor of electron is oxygen. Okay, and now we finished the glycolysis in cytoplasm. Now we went to the mitochondria, and in the mitochondrial matrix, we are present in the mitochondrial matrix, where pyruvate has to be further processed in this first step. So what will happen in the mitochondrial matrix is this pyruvate or the pyruvic acid here combines with coenzyme A, and combining with this coenzyme A, this enzyme helps pyruvate dehydrogenase to produce acetyl coenzyme A. And this acetyl coenzyme A then enters a cyclic pathway called the tricarboxylic acid cycle, or the TCA cycle, or the most commonly known as the Krebs cycle, after the scientist name Hans Krebs. So we produced pyruvate here. Now pyruvate went to the mitochondrial matrix by in the presence of my pyruvate dehydrogenase, got converted into acetyl coenzyme A, and in this process, one NADH2 and one carbon dioxide is produced. Carbon dioxide is irrelevant. Just you remember this, but remember this one NADH2, one NADH is produced. Which step? For pyruvic acid to acetyl coenzyme A conversion. Now, this, as I said, this acetyl coenzyme A will enter into the Krebs cycle. Okay, so we will see the Krebs cycle. So this is what the Krebs cycle looks like. Here is your glucose. The acetyl coenzyme A, once produced, will enter the Krebs cycle, and the first acceptor of Krebs cycle is oxaloacetic acid, OAA. And I'm this you have already studied. I'm not going to talk about each and every step and the enzyme involved. Everything is noted down here. You can have a look here for like remembering the steps here, like citrate to isocitrate, alpha ketoglutarate, succinate, fumarate. But here the important thing is the step where NADH is produced or where FADH2 is produced and how much or the number it is produced. So the step where isocitrate is converted to alpha ketoglutarate, see 1 NADH is produced. Similarly, when alpha ketoglutarate is converted to succinyl coenzyme A, 1 NADH is produced. And when succinyl coenzyme A is converted to succinate, one GDP is produced, which subsequently gets converted to one ATP. And when succinate gets converted to fumarate, look here, one FADH2 is produced. Similarly, when malate to oxaloacetate, one NADH is produced. Now you tell me how many NADH is produced. Here, one. Okay, I'll go to the pointer then. Here one NADH, here one NADH, here one NADH. And before this, for the pyruvic acid to acetyl coenzyme A conversion, one NADH. So in this step also, in this particular step, one NADH is getting produced. I'm just writing simply. So total how much of NADH? Yeah, anyone? For one pyruvate, how much of NADH? Okay, you are want the Krebs cycle. Okay, yes, correct. Four. So total four NADH. Now, in the previous, you saw one glucose molecule is giving two pyruvate. So one pyruvate is giving four NADH. So total NADH from one glucose is how much? Eight. Eight NADH. So the entire Krebs cycle is nothing just like what is Krebs cycle? Is Krebs cycle is happening in the matrix of the mitochondria. Uh, since it like what is the role of or what is the exact role of this Krebs cycle? It is uh, it takes the pyruvate and do the oxidation of the pyruvate. And in this oxidation of the pyruvate, the main goal of the Krebs cycle is to produce NADH and FADH and as well as little ATP. There's nothing to explain here in the Krebs cycle. It is a cyclic process where the acetyl coenzyme A is accepted by one molecule and it gets converted to another molecule by the presence of a particular enzyme. Like oxaloacetic acid or the oxaloacetate accepts the acetyl coenzyme A and in the presence of citrate synthase, it gets converted to citrate. You just have to look at the picture in the end of this class. I'll upload this PPT also and you'll have access to this. You just have to go through this. The main reason I'm showing you this one is that I want you to calculate yourself, not just memorize that you have to calculate yourself how one molecule of glucose is giving 38 more ATP. That is the main motto of my presentation of glycolysis. That how from one glucose molecule you get 38. Some people, somewhere you might find in some text or something 36, complete nonsense. One glucose will always give 38 ATP, and we will have the calculation here. Just remember how much ATP and NADH we still are left with. So at the end of glucose to pyruvate, we have two ATP, two NADH, and two pyruvate. Now pyruvate went in the Krebs cycle, and each pyruvate is giving three carbon dioxide, four NADH, one FADH2, and one ATP. So that means if one pyruvate is giving this much amount, two pyruvate, because one glucose was split to two pyruvate or two pyruvic acid. Then two of pyruvate will give how much? NADH 8, FADH 2, and ATP 2. I hope I'm clear till this step. I first started with glucose, ended with pyruvate. I showed what is the net ATP and NADH. Now I started again from the pyruvate, ended up in the Krebs cycle, and I showed you for two pyruvate what is the net output, how much NADH, how much FADH, and ATP. Okay. Now you will ask, what is this NADH and FADH2 is going to do? That is the next step of the energy production, where this NADH2 and FADH2 will be utilized in something called as the electron transport chain, which is not located in the matrix of the mitochondria, but is located in the membrane of the mitochondria. And which membrane? The inner mitochondrial membrane. This is the important thing to remember. Glycolysis is occurring in the cytoplasm. Pyruvate transported to the matrix of the mitochondria. Matrix, Krebs cycle happened. What you got? NADH and FADH. Now this NADH and FADH will be used in the electron transport chain or the electron transport system happening in the inner mitochondrial membrane. So electron transport system is to release and utilize the energy that is stored in NADH and FADH2. And this is accomplished when they are oxidized, and see, subsequent oxidation is going on. So this NADH and FADH is oxidized through the electron transport system, and the electron is finally passed to the last acceptor of electron, that is oxygen here. Oxygen resulting in the formation of water. But here, uh, remember this, like last time also I showed you the production of ATP by F0 F1 particle, or the ATP synthase, which is in the last here, and it is in the inner mitochondrial membrane. So I also told you that for one molecule of ATP, or one ATP, four hydrogen atom has to cross this. Okay, F0, F1 particle. Remember, this also comes in the multiple choice question. How many ATP? Okay, so now have a look at this, where the NADH is used and where the FADH is used. So electron transport chain is starts with the complex one. So there are many complex, or to be precise, there are four complex or four system which are active in the electron transport chain: complex 1, complex 3, complex 2, and complex 4. It starts with complex 1, which oxidizes NADH and mix and takes out electron from it. These electrons circulate from complex 1 to complex 4 via certain particles, or you can say molecules present in the inner mitochondrial membrane. And these molecules are divided into complexes like cytochrome C, or cytochrome A, this complex 4 having copper. So various name are given for the different complex. You don't have, like, you already read about this in class 12th. Uh, if you have not read, you will read these things in detail in a textbook. I'm not going to, like, this is beyond scope to discuss each and every complex, what is present in each complex. So you just have to, like, any standard text, you can have a look what is there in each complex. But the idea is the electron is extracted from this NADH and FADH. So from NADH, this electron is extracted by complex 1, and from FADH2 or the FADH, the electron is extracted by the complex number 2. And this electron are passed into this inner membrane space. So here, so this is the matrix, this is the inner mitochondrial membrane, and this is the intermembrane space, and this is the outer membrane. Outer membrane. So for here, you can realize that the hydrogen or the electron, as it passes through the complex from complex 1 to 3 to 2 and 4, protons or the H plus ions are uh, transported outwards and gets accumulated in this intermembrane space. And this H plus or the proton then cross the F0 F1 particle or the ATP synthase, and in during this reaction ADP is coupled with inorganic phosphate to produce ATP, which is the currency of energy. Here you should remember that one NADH gives rise to three molecules of ATP, and one molecule of FADH2 produces two molecules of ATP. This is the common conversion rule that one NADH gives three molecule of ATP, and one molecule of your FADH2. Just 1 second. And one FADH2 produces two molecules of ATP. So uh, we started with glucose, and now we just ended up having ATP. We had NADH2, FADH2, which are subsequently used in the electron transport chain, and all we have left is with ATP. Now the assignment is to calculate the total number of ATP produced. So now we'll write down what is the final uh, calculation, or how much ATP was produced in the glycolysis, how much was produced in the Krebs cycle, and using this conversion formula, we'll try to figure out how much ATP is there. So here in the Krebs cycle, there were two eight NADH, two FADH2, and two ATP. In the glycolysis, there were two ATP, two NADH. So what is the total now? NADH become 10, FADH become 2. See, I'm not telling something new. FADH2 become 2, NADH to 8 plus 2 become 10. And the ATP total 2 plus 2, 4. Here, I'll show you again. ATP2, here ATP. In the glycolysis, 2 ATP. Glycolysis, 2 ATP. Krebs cycle, 2 ATP. Total ATP, 4. NADH, in the Krebs cycle, 8. In glycolysis, 2. Total become 10. And FADH2, only in Krebs cycle, 2. So, now we ended up having the total calculation NADH 10, FADH 2, and ATP 4. Now all you have to do is calculate what is the total number of ATP produced. Since one NADH gives 3 ATP, multiply with 3, we will end up with 30 ATP. One FADH2 gives 2 ATP, multiply with 2, 2 to 4. ATP. ATP is remains ATP 4. So what is the total amount? 38 ATP. we started with one molecule of glucose, and at the end of the ATS or the electron transport chain, one molecule of glucose became 38 molecules of ATP, and this is all about the aerobic respiration. That's all. I hope you all are clear about the calculation of ATP and how the aerobic respiration is happening and the location where glycolysis, where Krebs cycle, where ETS. Correct? Everyone, are you clear about this?
Yes, sir.
Okay, okay, great. So we will quickly solve a few questions regarding this glycolysis. So the first one, the purpose of phosphorylating glucose during glycolysis is to trap the glucose inside the cell and commit it to the glycolysis, or to facilitate active transport into the cell, or to convert it into a disaccharide, or to produce energy directly. You can put it in the chat box also. So what do you think? Why this phosphorylation happens? Uh, what is the purpose of this glucose, like phosphorylating the glucose?
Sir,
So
option A.
option A, correct. So glucose can be used as some other purpose, like used for storage if it is not used. How to earmark it for glycolysis? So the first very step, this is this one. Glucose 6-phosphate. Use energy and add a phosphate tag so that it further proceeds in the glycolysis pathway. So that is the reason why the phosphorylation happens during the glycolysis, to trap glucose inside the cell and commit it to the glycolysis. So this step, it is committing to the glycolysis. So that is the reason it is option A. The next question, the role of NAD plus or NADH in glycolysis is serve as an oxidizing agent to accept electron, act as an reducing agent for pyruvate, or form the backbone of glucose, or convert ADP to ATP. Which one do you think NAD plus is working? Okay, so I'll quickly tell you this one. So it is option A. It serves as an oxidizing agent to accept electron. Okay, so I'll show you one here. See, NAD plus accepts the electron and gets converted to NADH. So I told you that these should undergo oxidation. So if oxidation, something should get reduced, like accept the electron. So this NAD plus is the uh, player. Uh, Serves as an oxidizing agent. Oxidizing agent is the like a molecule or the agent that can oxidize. So NAD plus acts as an oxidizing agent to accept electrons, and in the process gets converted to NADH. Hmm. Okay. Okay. So the NADH, the third question, the NADH that is generated during glycolysis under aerobic condition is dash. Stored in the mitochondria, enter into the electron transport chain, or inhibits glycolysis, or converted back to NAD plus by reducing pyruvate to lactate. during glycolysis? What do you think it is the fate of NADH? So NADH is used in the final step. And what is the final step? Anyone?
Sir,
Okay.
option D.
Option D. Oh, okay. No, okay. Converted back to, oh, we are talking about anaerobic condition. Okay, sorry. I thought it is aerobic condition. Yeah, correct. So the question here is anaerobic condition. And in this anaerobic condition, it is converted back to NAD plus by reducing pyruvate to lactate. So if it has been aerobic respiration, okay, this one I have to tell. Like if it has been aerobic respiration, then it would go to electron transport chain. Since in the question is there it anaerobic condition, there is two types of respiration, aerobic respiration and anaerobic respiration. So in anaerobic respiration, it is the quick release of energy. So this cannot wait. The NADH cannot wait to go through the electron transport chain, and then the coupling of hydrogen ions by ATP synthase, and then the ATP production. So for the quick release, we have the anaerobic condition, which produces less amount of energy as compared to the aerobic condition. So where this quick energy is required, uh, like when you are exercising, your muscle needs quick energy. You have this lactic acid pathway. From pyruvic acid, lactic acid is produced, and here the NADH is directly converted back to NAD plus by reducing pyruvate to lactate. And so that is the reason if you exercise more, then your muscle becomes sore or starts to pain due to this accumulation of this lactic acid. Similarly, for the pyruvic acid is also converted to ethanol in the process of fermentation by yeast. And that is where also NADH is used and gets converted back to NAD plus. Okay, correct. So under anaerobic condition, this option D is correct. Converted back to NAD plus by reducing pyruvate to lactate. Okay. So we are done with this glycolysis part. Now we will go to biomolecules. So biomolecule are the ingredient of cellular organelle and are also called macromolecule or normally called biomolecule. There are four major types of biomolecule. One is carbohydrate, protein, lipid, and nucleic acid. Nucleic acid uh, and protein and carbohydrate and lipid I'll discuss today, and the protein in the next part. So carbohydrate we'll discuss first. These are chemically the carbohydrate are aldehyde and ketone derivative of polyhydric alcohols. So to give you recap, this is what aldehyde looks like, CHO, and this is what ketone looks like, C double bond O. And the polyhydric alcohol are nothing but sugar alcohol, which are organic compound that contain more than one OH group or the hydroxyl group. So these are aldehyde and ketone derivative of polyhydric alcohols, carbohydrates. And the major role of carbohydrate in living organism is to function as primary source of energy. This is also known. These molecules serve as energy store, metabolic intermediate, and one of the major component of the bacteria and the plant cell wall. Carbohydrate is also used in the cell wall. So they are classified, the carbohydrate is classified into monosaccharide, oligosaccharide, and polysaccharide. We'll discuss about this in the next slide. So monosaccharide, the simplest carbohydrate are called the monosaccharides. The monosaccharide have free aldehyde and the free ketone group with two or more hydroxyl group with a general formula CnH2ON. This is the tabular form of the classification of monosaccharide. There are different classes of monosaccharide based on the number of carbon atom. If there is three carbon, triose, four tetrose, five pentose. These pentose sugar are important for the formation of nucleic acid. Then hexose, like glucose, the mainly used in the energy. So say like I told you there can be aldehyde group and there can be ketone group. So it can be classified as aldose and ketose based on whether it has aldehyde group or it has ketone group. Like for example, this hexose can have aldehyde group, so it will be called glucose. And if it has ketone group, it will be called fructose. Just minor difference and the name changes. And these are the monomeric unit or the simplest unit of carbohydrate, which are called monosaccharide. If we go to a little more complex, like carbohydrate having 2 to 10 unit of monosaccharide joined together, and the linkage is called glycosidic bond. So joining of carbohydrate, it is always glycosidic bond. Example of oligosaccharide, maltose, lactose, sucrose. These are the common example of oligosaccharide. If we go to the more complex carbohydrate, we will have 10 or more monosaccharide unit joined together by glycosidic linkage, and these are classified in a number of ways depending upon the type of repeating monosaccharide. Either they are homo polysaccharides or they are heteropolysaccharides, and they also differ in the degree of branching and the type of glycosidic linkage between the monomeric unit. Example are starch, glycogen, cellulose, chitin. All these structures I'm not putting here. This will make it very long and very confusing. You have already read what is the 1,4 glycosidic linkage in I think in cellulose and glycogen. So these are just you can go through these text and have a look. But these are the basic idea how they are getting classified into monosaccharide, oligosaccharide, and polysaccharide. 2 to 10 oligosaccharide, more than 10 monomeric units, they are classified as polysaccharide. So here I want to discuss about two more concepts in carbohydrate that for example, we will discuss about the glucose. So glucose has two types of structure. One on the left, it is called as the straight chain or the linear structure of the glucose, and on the right you have a cyclic form of the glucose. But how this cyclic form is producing or happening, it's because something called as the hemiacetal formation by intramolecular reaction between carbonyl group and the hydroxyl group. Between the hydroxyl group and the carbonyl group, there is intramolecular reaction, and that is what it makes to bend or fold like this, and this is called hemiacetal formation. So here alcohol group or the OH group is either reacting with aldehyde or reacting with the ketone. If it is reacting with aldehyde, then it forms a hemiacetal. If the alcohol is reacting with ketone, it forms hemiketal. Just look at this reaction, you will understand the the formation of a hemiacetal and hemiketal, and that is the reason why glucose exists as the either straight chain or hemiacetal structure of a cyclic form. So that is You remember this term hemiacetal, and we will use this in the next slide. There is also uh, like classification based on the rotation of the plane polarized light. You know this levo rotatory and dextro rotatory. But on what basis of or what is the difference, major difference that is used to classify the L and D form? So the orientation of the hydroxyl group or the OH group, that is most distance from the carbonyl carbon. That is most distance from the carbonyl carbon. And the orientation of the OH group, which is present in the most distance from the carbonyl carbon, determines whether the sugar belongs to D or L. So look at this image or the picture here. So the most distance of the carbonyl carbon, if this is the carbonyl carbon, the most distance from this carbonyl carbon is this particular carbon. Now, if the hydroxyl group is on the right side of the carbon atom bearing it, then it is called diisomer. See, if the OH group is present on the right side, it is called diisomer. When it is on the left side, the OH group, it is called L isomer. So that is how it is classified on into L and D sugar or L and D glucose by looking at the most distance from the carbonyl carbon. And most of the sugar present in the biological system, whatever we are dealing in the biological system, most of the sugar are D form of the sugar. So there's an interesting thing about like why the biological system has evolved to produce the D form and not the L form. So uh, kind of L form is mostly degraded by the enzyme. A lot of enzyme can degrade the L form. This is a what do you call that? Evolutionary conservation kind of concept. So most of the bacteria, not I can't say bacteria, or the enzyme which is present in the system, they are kind of mechanized to degrade these L isoforms rather than the D isoform. So if something is going towards glycolysis or if something is going to be stored, then the body generally prefers the D isoform. And this is also true about the most of the uh, biomolecule. If you are dealing with isomers, the biological system prefers the D form. Okay, so I wanted to say this that the biological system, the most common sugar are the D form of the sugar. Okay, so here we saw about the hemiacetal formation. Now, using that concept, there is another type of classification or another term which is called as anomers. So what is anomer? So isomeric forms of monosaccharide that differ only in the configuration about the hemiacetal or the hemiketal carbon atom are called anomer. So there you saw the hemiacetal formation here. Your first CHO reacting with your this fifth carbon and this this OH group and this CHO, because one alcohol will react with this aldehyde and it will form this kind of ring. See, From here it is the label is here so that you won't get confused. 1, 2, 3, 4, 5. Here 5 is forming ring with 1, and the 6 CH2OH is just like that. So since this carbon, the fifth carbon, is the hemiacetal carbon, depending on the orientation of OH group in the hemiacetal carbon, we have anomers. So if the carbonyl carbon atom, the carbonyl carbon atom is called the anomeric carbon. So this carbonyl carbon, as I showed in the previous one, the first one, the carbonyl carbon, is called the anomeric carbon. Now see, depending on the orientation of the OH group in the anomeric carbon, we have classification alpha and beta. How? In the alpha anomer, the OH group of the carbon is on the opposite of the sugar ring from the CH2OH group. So it is saying in the alpha, the OH group and the CH2OH are in the opposite direction. If this is towards the upside, OH is towards the downside. If you find this type of structure in a hemiacetal structure, like the CH2OH is towards one side and the OH in the hemiacetal carbon or the carbonyl carbon are both in the opposite direction, then it is alpha. If it is in the same side, the OH in the hydroxyl group in the carbonyl carbon or the anomeric carbon is towards the same side as the CH2OH, it is called as the beta form or the beta anomer. So based on this um, two types of orientation of OH in the anomeric carbon, we have alpha anomer and we have beta anomer. So now you can differentiate between alpha D-glucopyranose and beta D-glucopyranose. Pyranose, this structure, circular form of sugar is called this ring type of structure is called pyran structure. That is why it is glucose and it is circular in structure. That is why it is called glucopyranose. But I want you to understand why it is written as alpha D and why it is beta D. So D and L I discussed about here. Alpha and beta I discussed here in the concept of anomers. You should remember this anomer. Okay, so like not very complicated. You can draw it by yourself and then can simply remember this. Apart from anomer, we have another uh, term called as the epimer. So in epimer, we have the same molecule like glucose has two different names. One is galactose, one is mannose. And why? Because isomer having different configuration of OH or the hydroxyl group only at a single carbon atom are known as epimer. The most important epimer of glucose are mannose, which is different in the like the carbon atom number 2. See the mannose and the glucose. See the carbon atom 2. The OH group has been changed, or the orientation of the OH group has been changed. And glucose and galactose. Look at the carbon atom 4. 1, 2, 3, 4. So this is look at here and look at here. If there is difference in the like the OH orientation in a single carbon atom, they are called as epimer. Okay. So that's all for anomer and epimer. So based on the like epimerization of the glucose, you will have mannose and galactose. So just like take your time and then like if you memorize the concept on the concept how this classification is happening, it will be very much easier. There's nothing much. One in one case it becomes cyclic, and in another one we have different names based on the orientation of the OH group. Okay, I hope this part is clear. We will go to the questions then. Okay, I hope everything is like clear. I'm just telling you simple concept. If you want to ask anything, you can ask, or I'll go to the questions. But after the end of this, download this PPT and like write yourself, draw the structure yourself, and this will be like very clear to memorize. Anomer, epimer. Okay. So we will go to the questions using these concepts. The first one, the group of monosaccharide that is primarily used for energy in metabolism. Ketopentoses, amino sugar, aldohexose or deoxysugar. So it is between option A and C. Pentose or hexose, which go for energy. In glycolysis, what is the number of carbon atom? Anyone? So I'm telling you, it is between option A and option C. So forget about this keto and aldo. One is pentose sugar, one is hexose sugar. So which is generally preferred for energy. So glucose is a hexose sugar. It is a six carbon. Hexose means six. So pentose is preferred in the case of nucleic acid formation of nucleotides. So the option is option number C, aldohexose. Aldo is just the aldehyde group is there. Ketohexose can be there. Both aldohexose and ketohexose can go for the energy metabolism, but glucose is an aldohexose. Okay. So the next question. The characteristic feature that differentiate beta D-ribofuranose from alpha D-ribofuranose. Note here, you can answer this question yourself. Both are D-ribofuranose. The difference is beta and alpha. One is beta D-ribofuranose, one is alpha D-ribofuranose. So, which is it? Orientation of hydroxyl group on anomeric carbon, or the number of carbon in the molecule, or presence of ketone group, or configuration of alkyl carbons. Yeah, people, can you like this is very simple. Or do you want me to repeat this concept?
Sir, option A.
Okay, correct. Yeah, it is option A. So, as I said here, in the case of anomer, beta and alpha or alpha and beta classification is done on the basis of orientation of OH group in the anomeric carbon. So, since in this question there is both D-ribofuranose and D-ribofuranose, only difference is beta and alpha. It is automatically understood that we are talking about the orientation of hydroxyl group on the anomeric carbon. In exam also, you use this trick to answer this. See, only if it had been L and D, that is different. But here only beta and alpha is different, and that means we are talking about anomers, beta anomer and alpha anomer. So that is the reason why it is option A. Next question. The type of reaction that forms a hemiacetal structure in carbohydrate is dehydration of a sugar molecule or condensation of two sugar or the reaction between two ketone group or the reaction between aldehyde and the alcohol group with the same molecule within the same molecule. This one also I told you, formation of a circle is by the reaction of either hemiacetal or hemiketal. Okay, so it is option D, hemiacetal. He- Hemiacetal is aldehyde reaction with alcohol group. I'll show it one more time here. Here, hemiacetal. Alcohol group reacts with aldehyde to form this type of ring structure is called hemiacetal. This alcohol group reacts with ketone, forms hemiketal, and this is called hemiacetal formation. And this is the reason why glucose exists as linear chain as well as in the cyclic form. Okay. So this is the option D, reaction between aldehyde and alcohol group within the same molecule. Remember, this is also important that it is within the same molecule. Okay. The next question, the sugar that is the ketose analog of glyceraldehyde. Does anyone know this question? Like glyceraldehyde is the smallest monosaccharide. What is the ketose analog? So this is the aldehyde analog. What is the ketose analog of glyceraldehyde?
Sir, option B.
Option B, correct. So the simplest ketone analog is the dihydroxyacetone. So on the in the left you can see this is the glyceraldehyde and which has a CHO group, and the simplest monosaccharide for a ketose analog of is the dihydroxyacetone C double bond group. Okay. The test that is commonly used to identify reducing sugar. So we have two types of sugar. One is reducing, one is non-reducing. Anyone done the experiment in their school? Identify reducing sugar. Is it biuret, Benedict,
Yes, sir.
iodine? Sorry.
Benedict, sir.
Yes, yes. So Benedict, commonly Benedict test is used to identify reducing sugar. So, as I said, carbohydrate may be classified as reducing or non-reducing sugar. All those carbohydrate which reduce failing solution or the tolerance reagent are referred to as reducing sugar. So reducing sugar, where the anomeric carbon has OH group attached, can reduce other compounds. Uh, Non-reducing sugar do not have the OH group attached to the anomeric carbon, so they cannot reduce other compounds. So reducing sugar is some like the class of sugar which has the free OH group. Since they have a free OH group, they can reduce other compounds. Since we discussed all in the section of monosaccharide, that monosaccharide has a free OH group, irrespective of whether it is aldose or ketose. That is the reason why all monosaccharide, since they have free OH group, are reducing sugar. Any monosaccharide, irrespective of aldose and ketose, they are called they are called reducing sugar. And this is all about the Benedict's test and the reducing sugar convert the cupric ion or the Cu2 plus in the reagent, Benedict reagent to cuprous ion or Cu plus. The copper ion then forms copper 1 oxide, which is precipitate as a brick red color. And this is a test to identify the reducing sugar. Okay, so this is the Benedict's test. The second part, we are done with the carbohydrate. In the second part, I'm going to tell you about the fatty acid and lipid. So, fatty acid are obtained like these are nothing but fats and lipids. So, fatty acid are the simple one, and then the complex of this fatty acid are lipids. So fatty acid are obtained by the result of hydrolysis of fat, and they are naturally occurring fatty acid are generally synthesized from two carbon units and hence contain even number of carbon atoms. So like there's a long chain CH2 CH2, 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 CH2. So there is even number of carbon atom, and in the end, you can see this type of structure: C double bond O and COH. And this is a typical structure of fatty acid, and this in particular is stearic acid. Synthesized from two carbon unit fatty acid chain may be, and as I said, synthesized from two carbon unit, and they are hence they are even number of carbon atoms are present. Fatty acid may be either they are can be saturated. In saturated, there is no double bond or unsaturated. Unsaturated have one or more double bonds. Uh, this one you should like no, because when you look at the fatty acid in a table, they don't write sometimes they don't write the name, or they sometimes do not draw the entire structure because these are long structure. Then how to understand how many carbon are there, how many single bond or double bond are there, and where exactly the double bond is there. Hmm. Sorry. So this is the writing style of the fatty acid, and they are denoted by the total number of carbon followed by a colon, and then the total number of double bond with a delta symbol having superscript number defining the position of the double bond in thesis. For example, what I told you, let's say there is 18 carbon long fatty acid with two double bond between carbon 9 and carbon 10 and carbon 12 and carbon 13. How you will write this fatty acid? So it 18. So first you will write the total number of carbon atom 18. Then you give a colon mark, that is colon, and then you will put how many double bonds are there. So it says there is two double bonds. So you will write 18 is to 2. Then you will start the bracket, and in the bracket you will put delta, and you will write what is the position of the two double bonds between carbon 9 and between carbon 12, like 9 and 10 and 12 and 13. So you will write 9 comma 12. If there has been another double bond between carbon 15 and 16, you would have written 18 is to 3 delta 9 comma 12 comma 15. So this is how you write or like give an annotation to the fatty acid. Okay. Oh, okay. I hope you are clear about this one. So I was talking about the unsaturated fatty acid. So unsaturated, like saturated fatty acid do not have any double bond, You don't have to like worry more about that. The unsaturated fatty acid have one or more double bond. So we need to classify further the unsaturated fatty acid. They are further classified into either monounsaturated fatty acid or the MUFAs or in the polyunsaturated fatty acid or the PUFS. So monounsaturated fatty acids contain only a single double bond, and the PUFS contain more than one double bond. And this is a list, entire list, which with the common example of the saturated and the unsaturated the fatty acid. And like in the case of unsaturated fatty acid, here is the example, like the style of writing. For example, uh, in linoleic acid, it says 18 is to 2, bracket delta 9 and 12. That means it is a 18 carbon with two double bond in the between carbon 9 and 10 and carbon 12 and 13. Example, arachidonic acid, 20 is to 4. That means 20 carbon atom having 4 double bond between carbon 5 and 6, 8 and 9, 11 and 12, 14 and 15. So by just looking at this, you can tell how many carbon are there, how many double bond are there, and the location of the double bond in the case of unsaturated fatty acid. In the case of saturated fatty acid, since there are no double bond, everything is 0. 12 is to 0, 14 is to 0, 16 is to 0. So I hope you all are clear about unsaturated fatty acids, saturated fatty acid, and the writing style of the unsaturated fatty acid. Okay. Okay. Now we will go to the lipids. So lipid is something more complex of the fatty acid. So lipid are primarily made up of hydrocarbon chain connected to glycerol via ester linkage. So this is a ester here. C double bond O R dash. This R and R dash are different. So hydrocarbon chain connected to glycerol via ester linkages. These are the lipid. And we broadly classify lipid into two categories. Either you classify to simple lipid or you classify to compound lipid. Various type of lipid are included within these two major categories of lipid. These include fat, triglycerol, wax, phospholipid, and steroid. We will just have a brief look at the simple lipid and the compound lipid. So simple lipid are the fatty acid having various alcohol with no other group. And simple example is fat and wax. And as I said, this is a ester, like the esterification. So the first simple lipid is triglycerol or the triglyceride. Most common one, commonly known as the triglyceride or neutral fat, are compound. These are the compounds are the ester of glycerol and fatty acid. You can see that the glycerol part is this one. This is the glycerol part. Okay, you all know like I believe that you all know for the structural formula of glycerol. So this is the east like the see this is the ester, glycerol, and then the chain, the fatty acid chain. This is the fatty acid chain. This is your ester. This is your glycerol. So this is what it is written here. It contains the same fatty acid. The same fatty acid in all the three ester position are called simple triglyceride. Okay, I'll just maybe draw this for you. This one. If these three are the same, this is called simple triglyceride. If the triglyceride contain more than one fatty acid, if like let's say these are not same, there is more than one type of fatty acid, then these are called mixed triglycerides. Triglycerol is essential. Remember this one, triacylglycerol or the triglyceride is essential for providing energy to the body. Triglyceride are the vehicle of energy storage primarily in the adipose tissue. These are stored, triglycerides are stored in the adipose tissue for the energy storage purpose. Another one of simple lipid is wax. So wax is formed as a result of esterification of fatty acid with monohydric alcohol of higher molecular weight. So here the alcohol is of higher molecular weight monohydric alcohol. Wax are found in a wide variety of organisms. For example, wax serves as a protective coating on the skin of animal and plant and also reduces water loss in the tropical plants. You like may not remember what is the like these complex terms like esterification of fatty acid with monohydric alcohol, but you should at least remember in simple lipid what is the members like triglyceride and wax are the simple lipid. And triglyceride you can easily remember this structure because this is a very easy structure to remember. The simple triglyceride. Okay, so now we'll go to the compound lipid. So in the compound lipid, are fatty acid ester with alcohol. Here some additional groups are there. We will see what is this additional group. So the fatty acid component of this lipid make the hydrophobic tail and the alcohol moiety along with the additional group constitute the hydrophilic head. You can already imagine what I'm talking about. Hydrophilic tail, additional moiety forming the hydrophilic head, like a phospholipid in phospholipid bilayer. So here the additional group there is a phospholipid, and that is why it is called a compound lipid. Such a structure, this having hydrophilic head and a hydrophobic tail, the structure is called an amphipathic molecule, and common example of compound lipid are phospholipid and the steroid. So as like on the left, it is for their like discusses about phospholipid, right? Discusses about steroid. See, this is what I was telling you about the lipid. In the phospholipid, we have two types of phospholipid, either glycerophospholipids or the sphingophospholipids. So the phospholipids are primarily found in the cell membrane. In the fluid mosaic model of plasma membrane or the cell membrane, I already told you about this. These compounds are composed of fatty acid chain. This is the fatty acid chain, like a saturated fatty acid, unsaturated fatty acid here attached to a glycerol backbone. This is your glycerol backbone, and this is your additional group, which is a phosphate group. And if that instead of this phosphate there is sphingosine unit, it becomes sphingolipid. Sphingolipid in your neurons, the coating of your neurons. So I hope you understood this phospholipid. Coming to the steroid are distinct from other lipid in having a peculiar four fused ring structure. See, one, two, three, four. Four fused ring structure. However, like all other type of lipid discussed so far, steroid are hydrophobic in nature and insoluble in water. The steroid are strictly hydrophobic in nature and insoluble in water. In a cell, steroid act as a receptor ligand and help to control the metabolism. You will see like how it acts receptor ligand and control metabolism. Like example, most common is the cholesterol is the common derivative of steroid, and it is the key precursor of all steroid hormone, including testosterone, estradiol. So steroid are mainly used for the hormones. Okay, so this is the structure of cholesterol having typical four fused ring structure. Okay, so I'm done with the liquid part. Now some few question, and then quickly just 1 2 slides for nucleic acid, and we'll be done. So I hope you people are clear with the lipid part, the fatty acid and the lipid part, and the classification of the lipids. Okay, so answer these questions quickly. The lipid type that is commonly found in the myelin sheath of neuron is triglyceride, sphingolipid, steroid, or wax.
Option B, sir.
Option B. Very easy to answer. Sphingolipid. So option B. Just a second. Okay. Next question. The lipid class that serves as a precursor for hormones like testosterone and estrogen is triglyceride, phospholipid, steroid, or wax. So
Option C.
option C. So here I told you about the steroid, which are mainly in hormones like testosterone and estradiol. So it is option number C. The storage form of lipid in animals stored in the adipose: phospholipid, cholesterol ester, triglyceride, or glycolipids.
Option C, sir.
Option C, triglyceride stored in the adipose. The next one, the unsaturated fatty acid affect the membrane fluidity because unsaturated fatty acid has what? They increase the rigidity by forming tight packed structure, or form kinks in the hydrocarbon, or prevent interaction with cholesterol, or have no impact.
Sir, I think option B.
Option B. So they decrease the rigidity by introducing kinks, these double bonds in the hydrocarbon chain, and that is how they affect the membrane fluidity. So that is option number B. Okay, the next question. The structural feature that distinguishes saturated from unsaturated fatty acid is very simple to answer. Length of the carbon, or the type of functional group, or the solubility, or the presence of double bond.
Option D, sir.
Option D, presence of double bond. Okay. So now nucleic acid. This is very widely discussed. I think all of you know about this nucleic acid. So these are the polymer of nucleotide linked through phosphodiester linkage. So that is glycosidic linkages in case of your carbohydrate. This is phosphodiester linkage in case of nucleic acid. Two type of nucleic acid are present in the cell, namely DNA, deoxyribonucleic acid, and the RNA, ribonucleic acid. In our case, DNA is the genetic material and inherit the information from one generation to the next. In case of virus, RNA, like many virus, there are also DNA viruses, but most of there are RNA viruses, which use reverse transcriptase to hack the like once inside the host, they use reverse transcriptase, form their own DNA, and then hack the DNA mechanism of the host. So, bottom line is RNA is the like genetic material of many viruses. If there is question that what can act as genetic material? Only DNA, only RNA, both DNA and RNA. So both DNA and RNA can act as genetic material because RNA is the genetic material of some many viruses. So nucleotide of DNA and RNA. So what do you mean by the nucleotide? It is nitrogenous base plus sugar plus phosphate. Sugar, I already told you, pentose sugar. Here, pentose sugar makes up the nucleic acid. So two types of pentose sugar based on the OH group. Either if it is present, then it is called the D ribose. If in the 2' position, instead of OH only H is there, it is 2' deoxyribose. ribose and deoxyribose. And there is nitrogenous base. So nitrogenous base, you all know that we have purines and pyrimidines. Purines are adenine and guanine, and pyrimidine are uracil, cytosine, and thymine. So this nucleoside and nucleotide concept. So nucleoside is pentose sugar plus the base. Any base from here makes the nucleoside. Now in the 5' position, add the phosphate group also. Nucleoside will become nucleotide. That is a major difference between nucleoside and nucleotide. Nucleoside is the pentose sugar plus base. Pentose sugar plus base plus phosphate group makes it the nucleotide. So other concept is the phosphodiester linkage, and you all know this that the phosphodiester linkage is between the 3' OH group of the sugar of one nucleotide and the OH group of the phosphate attached to the 5. So many a time it is written 3' 5' phosphodiester linkage. And here during this condensation, one molecule of water is lost to form this phosphodiester linkage. Okay. So the question here is, what is the role of Chargaff's rule in the DNA structure? So the scientist Chargaff, he gave a very simple rule. So what does it predict? It predict the sugar phosphate backbone, or it determine the sequence of the base, or it establish the base pairing rule, A double bond T and the G triple bond C, or identify RNA's single stranded nature.
Option C, sir.
Option C. So Chargaff's rule is A finds the number of A adenosine and guanine, because if you find adenine, equal amount of thymine will be there because adenine double bonds with thymine. And if you know how much guanine is there, you can figure out how much cytosine is there because G triple bonds with C. So this is what Chargaff's rule is. He gave two rule. One, that the number of guanine is units equals the number of cytosine, and the number of adenine equals the number of thymine. And the rule number two is the composition of DNA varies from one species to another. Whatever the amount of ATGC is there in one species of animal is not equals to the another species, because in each species they have a different length of the DNA. So, and the base pairing rule, that A always pairs with T double bond, G always pairs with C. Okay. So here you might be wondering, what is this approximately equal? In general case, Chargaff's rule says that there is equal amount of A and T. Like if the DNA has some mutation or has undergone some like damage or repair, because at a given point of time, DNA has some minute mutation, some minute like damage or some repair might be going on. So at that particular point, A might not be pairing up with T, and on the opposite, there might be something else. Like in case of mutation, there might be a G in opposite of A. So there can be some minor variations, but that is the reason why this approximate word is used. Okay, due to this mutation, some minor DNA damage or DNA repair mechanism, which generally goes throughout the DNA. Okay, the last question and the last concept about the very wide known sequencing of DNA, or the method where the modified nucleotides are used to terminate DNA strand elongation. Maxim Gilbert, Sanger, Southern blot, or PCR.
Sir, I think option B.
Option B. Okay, correct. Most widely used in the past, and like we don't generally use the Sanger sequencing or the chain termination method. So it is option B. Currently, we use the NGS, next gen sequencing, and this is very old method, this Sanger sequencing, but it is most like very popular method of DNA sequencing. So in this, before ending, I'll quickly tell you about this Sanger sequencing. Like you people have already read about this. So this is called chain termination method. So chain termination method means for let's say any PCR, you need a template, you need a primer, you need DNTP, and you need a polymerase enzyme. So the polymerase enzyme will read your template and incorporate the DNTP in accordance with ATGC. Like if in the template A is there, this will incorporate the D. The who will incorporate the DNA polymerase. But in Sanger sequencing, in mixture, in addition to the DNTPs, you will take a unique mixture of DDNTPs. DDNTPs are DNTP are deoxynucleotide triphosphate. In DDNTP, these are dinucleotide deoxy triphosphates. So what will happen? Uh, in case of DDNTP, once a DDNTP is incorporated, the elongation of the chain cannot proceed further or the chain termination happens. So this is the pictorial representation of the Sanger sequencing, where you will take the template, make it single strand, take the primer and add a mixture of DNTP and DDNTP. And based on the incorporation of the DDNTP, your chain termination will happen. Like, see, this is what it is being read. Like, if it is ACTG, see, ACTGA is the primer. Then after that, it is A. One DDNTP is incorporated. So the chain terminated here. In the another fragment, like normal A got incorporated here, but just after that, one DDTP got incorporated here. So the chain terminated here. In another one, so this is kind of chance event, and then depending on where that DDNTP is getting incorporated, you will get different length of the fragment. And if you run this on an agarose gel, or we call this as a Sanger sequencing gel, you will get this type of band, because for each DDNTP you are using, let's say, a different color. So now if you run this agarose gel, you will see different size of band, because there are so many fragments got terminated like randomly by random incorporation of DDNTP, and they have different length. So you will agarose gel, you will see different size, because that is the property of agarose gel. It provides sieving effect, and they will be different fragment will be separated based on their size. The largest size will be at the top, the smallest size will be at the bottom. Now there is different color like assigned to the uh, like DDNTP. And if you align them in one line, and if you read them, then you can read what is the sequence of the DNA, like GATC. And since this is a very complicated, like you incorporate DNA, then you run the gel, then you interpret this gel. This used to take a very long time. So this is the concept of Sanger sequencing. Okay, so that's all for today. I am done with this part. So if you people have any question, feel free to ask. Otherwise, you are free to leave the meeting. Okay. Okay, people. So that's all for today. If you have any question, you can ask, or I'll close the session for today. Thank you all for joining.
Thank you, sir.
Thank you. Okay, thank you all.